downer? Okay.
Yeah, exactly.
So this is the computational analysis on receptor kinase NTRK and the mutants that we found, G595R and G667C, using virtual screening of an FDA-approved drug, Entrectinib. So how these two things work, so NTRK is involved in the development and maturation of the central and peripheral nervous system through the regulation of proliferation, differentiation, and survival of the sympathetic and nervous neurons. So it's a tumor-causing agent. It causes brain cancer. And Entrectinib is a selective inhibitor of the tyrosine kinases and tropomyosin receptor kinase, and has been proven to be effective against primary and metastatic brain diseases. So it is effective against the wild type. It has two drug-resistant mutants, which are the G595R and the G667C, and the IC50 is significantly higher with these mutants compared to the wild type, meaning it's more drug-resistant in the mutants. It's the most prevalent cancer from these mutations is thyroid cancer. And it's important that the new FDA approved drugs can be found that can combat these mutants as well as the wild type. And that's going to be discovered on the and hopefully further studied. And new drugs were discovered over numerous steps of computational analysis. So this is just kind of an overview of what we did over the past weeks of dry lab. So first we did a database search, which basically gives us information about the structure and function of our kinase. That's how I discovered that it was involved in the metastatic brain cancer. And then we found the protein structural properties, which we got an introduction to master and gave us a look at the structure, and homology modeling also gave us a 3D model of our protein. And then we were able to do docking, which allowed us to see the binding affinity of the wild type and its mutants and gave us the docking scores. And then once we got that information, we were able to do the virtual screening, which allowed us to see, for this one, there was 5 different FDA-approved drugs that were found and worked best for wild type and our mutants, and those could be further studied. So this is just another overview. As mentioned earlier, it shows our proteins involved in the development and the maturation of the central and peripheral nervous systems. And this is just kind of the stuff we got when we did the database search. And then we were able to get the protein structure. So on this side, on the left, this is just like the general structure, and this is just another view of the like the polarity of the protein. And then we did the docking, and this is where we got our docking score for the protein. And then this is what the ligand looked like when we did the docking score. And then when we did the virtual screening, this is when we were able to find the 5 different FDA-approved drugs. So this is the docking scores and the name and structure of each of them for the wild type. So you can see I'm not even going to try to pronounce all these, but these are the 5 different proteins, or 5 different drugs and their structures. And you can see their docking scores are usually around like -9.5 to -8.6 for the wild types. And then if you see there's a DBID, so this is how I compared these two. So you can see that the docking scores are a little higher for the mutants, meaning that the drugs were able to bind better to them, meaning these would probably be a better model for, or better drugs for the mutants. So for the research plan utilizing these different FDA-approved drugs as well as the Entrectinib, the best approach would be to test these drugs against each other and compare the IC50 scores of each with the 2 mutants and the wild type. This could be done using something we did similar to in the wet lab with the EGFR, where we use a kinase flow assay kit and measure the luminescence at 3 different concentrations of the inhibitor. And since kinases produce ADP from ATP, ATP will appear in luminescence. So if the more luminescent means that it wasn't able to convert that ATP to ADP. And this assay can be done for the drug as well as the 5 FDA-approved drugs that we were able to find in the virtual screening. So this is kind of what the graphs I got for the EGFR lab that we did. So this is kind of similar results that I would expect for this protein. So based off the docking scores from virtual screening, the IC50 should be lower for the 5 FDA drugs with the mutants rather than the wild type, because they were able to dock better, meaning that it'd be less drug resistant to these drugs. It's a good thing because it can better understanding of the mutants and what drugs can inhibit it. The IC50 should be lower with the Entrectinib and the wild type rather than with the mutant, because it's known that it works better on the wild type, and the mutants are more drug resistant to that. We want the IC50 to be low for both the mutants and the wild type, because it means that these drugs can work to inhibit the cancer causing kinase. So It's so dry right now.
Water. Just explain why that's confusion.
Yeah, because like I said earlier, it's like expected because we know that drug works on the wild type and that the mutants are more drug resistant. So the lower the IC50 score, the less drug resistant it is to the drug.
Okay, so my time we go back to the title.
The title?
Title. Okay, so really this is the A work, but we need to change the title. This title will confusion.
I didn't know the
Okay,
title.
so computation analysis. This is about from this people saw the you analyze how this drug works. That's not the purpose of this study, right?
Yeah, it's to find that. Yeah.
Find a new drug to replace this one.
I thought it sounded smart by putting computation.
You have to make it explicitly. So that's basically you change the title and rephrase. Don't confuse the audience.
Okay, because that's what I named my report, so I'll change that.
Yeah, change that one. So really, you're looking for the new compound to replace the existing drug.
Mhm.
So you have all the data. Yes, just that.
Okay.
Okay, so next one.